I love this slide because it reminds me of my grandchildren. Now, this is a healthy cell. And in every cell in our body, there is a factory called the mitochondria. And the mitochondria is critical for our energy levels because it produces adenosine triphosphate. Now, ATP accounts for 95% of our energy. So, in a young, healthy, robust child, you're going to have a cell that looks like that. And the cell shows a huge output of ATP and a small amount of free radical production. So, no wonder those grandchildren have the most unbelievable amount of energy and run around like crazy all day. Now watch my cell. I don't know what happened in the meantime, but notice what's happened. ATP production becomes very low and free radical production becomes very high. So we have to think about that in our prevention of pathology in terms of biochemistry. Okay, Kev? Very interesting Nobel Prize given in 1995 to Professor Günter Blobel. And Professor Günter Blobel discovered that proteins have intrinsic signals that govern transport and localization in the cell. What a brilliant step forward this was, because in a sense it actually explained our process. And what Professor Blobel found out is those signals which are attached to the sequence of amino acids are the signals that govern the direction and transportation in the body. Okay. Another very interesting professor in America, Professor Thomas O'Halloran, discovered that in a sense it was similar, but that proteins act as chaperones in the body. And as they act as chaperones, they take the nutrient to the destination that is required of that particular nutrient. And Professor O'Halloran did a most amazing amount of work in terms of copper. And he showed how copper, when it is attached to food, is chaperoned by the protein attachments to where it's supposed to be in the body. And this is a graphic representation of the nutrient bonded to the correct protein chaperones, now attached and bonded, and with a destination in mind, as opposed to an isolated nutrient with no transportation mechanism. So it has to find its own way. There are two basic foundation stones to the bonding of the nutrient to a base. And one of them is yeast, and one of them is your lactobacillus, your yogurt culture. Now, what you would want to do, therefore, is you'd want to, for example, bind your calcium to the yogurt, wouldn't you? Because you'd get extra benefit. The yeast, you would want to bind your seleniums and your zinc because you get magnificent benefit binding it to that yeast. Now, these are not the only two bases. We use other bases as well. For example, on vitamin C, we use a citrus pulp, a citrus combination. And so we always use a base. Okay, Kev? The interesting part of this new single cell plant grown nutrient, which gives us cellular nutrition now, a key word, is that it has this targeted delivery system. Okay, Kev? Okay, here's a very important point. People say, why do you give such low doses? Well, we give doses that are relevant. Now, just a simple example. The elephant, the African elephant, requires about 1.5 grams of calcium a day. But that elephant gets its calcium from the green grass that it eats. And it's bound to a food, bound to protein, and it supplies him sufficient calcium. We prescribe to the osteoporotic, especially ladies in our world, we prescribe 1.5 grams. Sometimes even 2 grams of calcium. And what's happening? Well, osteoporosis is increasing. Something going wrong there. Okay. Just some toxicity studies, and these are all boring, but I just wanted to show you that a lot of research has been done. This is toxicity studies relating to selenium and chromium, and the USP is always represented as 1 in terms of the nomenclature, but you can see that the toxicity, the toxicity of food state versus the USP is quite significant. Okay, Kev? This is the bioavailability of certain of the food matrix elements now in the liver, and you will see that the USP is related to 100 and the different amounts of bioavailability in the liver from the food matrix. Okay, Kev? Here's another study, and this is now bioavailability not in the liver but in serum, and you will see that relating to USP there is quite a large differential, and especially in terms of vitamin B12. And those of us in practice know that vitamin B12 is a fairly critical element that we are using more and more, and especially with the elderly people. Okay, Kev? This is the comparative bioavailability of a number of substances: food matrix, USP, gluconates, and chelate. You can see that the chelates actually performed pretty well, but best of all was the food matrix. So these are all university studies that have demonstrated the bioavailability of the food matrix. Okay, Kev? Now, just for a minute, calcium. When we prescribe calcium, we want calcium to benefit the bone. Now, all calcium is not the same. And the next slide will show that, okay, here's the point. The prescription that we give today is mostly calcium carbonate or calcium gluconate. There is huge deposits in the artery, in the kidney, in the gut, in the breast, and the prostate from the calcium carbonate. And the reason for that is that it is a rock, difficult for the body to process, and causes occlusion in our arteries. Okay, Kev, let's go. That's an occlusion picture for us. Okay, let's go. Calcium carbonate is limestone, calcium phosphate is chalk, calcium sulfate is plaster of Paris. Now those rocks are absorbed between 2 and 10%, so there is some absorption. However, there is poor membrane permeability, and so instead of getting it to the right place, we are depositing it in the wrong places. And food matrix calcium is absorbed at a much higher percentage, and it is, of course, more desirable because it doesn't leave depositions in different parts of the body.